Good morning, everyone. Have you ever wondered if there are any other ways to grow chili peppers? There are other ways to manipulate the growth of chili peppers so that there are variety of them. One of it is through propagation, but there is a twist to it, which is growing it in a small tissue culture, better known as in vitro. Before I get into the poster presentation, I would like to introduce myself. My name is Claudia Moau. I am a second year student from Resource Biotechnology. Today, I will present about the in vitro propagation of chili peppers, or scientifically known as Capsicum frutescens. Before I would like to start, I just want to say welcome to my presentation on in vitro propagation of chili peppers. On today's agenda, I will be delivering the information about chili peppers, such as the problem statement of this research. What is there to know about chili peppers, the hypothesis, objectives, experimental designs, and the expected outcomes of this research. And this is how the poster would look like. The problem statement of this research is that effective protocols for in vitro tissue culture and plant regeneration are required for the application of biotechnological tools like asexual reproduction of elite stocks, recovery of useful somaclonal variants, germplasm preservation and the production of transgenic plants with improved agronomic traits, interspecific hybrids, and haploid plants. Although numerous protocols and morphogenic methods have been used to accomplish in vitro regeneration of chili peppers, it has been challenging to develop effective, dependable, and broad-spectrum procedures for members of this genus. As a result, dependable regeneration methods for chili peppers are required, particularly for cultivars produced for commercial purposes. In this slide, I would like to introduce what is a chili pepper. As an introduction, chili pepper is a widely used ingredient in food preparation around the world, as well as a significant commodity in medicinal, food, cosmetic, and poultry industries. According to Ochoa Alejo and Ramirez Malagon in 2001, they are often used as a model system due to their ability to regenerate organs and eventually whole plants in vitro, as well as their ability to genetically modify using currently available transformation methods. In vitro micropropagation and genetic transformation have also been accomplished using organogenic systems. The use of cell and tissue cultures to select variant cells with increased tolerance to abiotic stresses have been effective, but no plants with the desired traits have been regenerated. The hypothesis of this experiment is to test that the whole life cycle of chili pepper plant can be completed under in vitro condition in a small tissue culture container. Whereas the objective of this experiment is to compare various media formulations, explants, and Capsicum frutescens germplasm sources for inducing in vitro morphogenic responses and complete plant regeneration. Other than that, this experiment is also carried out to equate the agronomic performances of regenerated plants with those of plants grown from seeds in a greenhouse. The method of this experiment starts with obtaining seeds from different species of capsicum. The in vitro morphogenic responses of three sources of explant, the cotyledons, hypocotyls, and zygotic embryos, were evaluated. Seeds from the various species were surface sterilized by immersing them in a solution containing 1 gram of chlorothalonil tetrachloroisothalonitrile for 5 minutes. The seeds were then immersed in a 30% volume-to-volume NaClO solution with 1.0 ml twin 20 for 20 minutes before being washed for 4 times with sterilized distilled water. Disinfected seeds were placed on half of maraschino and scooped medium with 1% weight to volume sucrose and solidified with 0.6% agar-agar. Whereas for the induction of morphogenic responses, the cotyledons, hypocotyls, and embryos were placed on 3 petri dishes with 30 ml of induction media, which were then labeled as medium A, medium B, and medium C. The induction media included the MS medium's basic salts, 3% sucrose, 11.8 micromole silver nitrate, 0.7% weight to volume agar-agar, and the following concentrations of N6-benzyl adenine and indole 3-acetic acid, which is stated in the slide. And for the elongation of adventitious shoots, the explants were transferred to glass jars containing 30 ml of elongation medium, which included the salts of MS medium, 3% sucrose, 2.9 micromole gibberellic acid, 23.5 micromole of silver nitrate, and 0.7% agar-agar. Next is the routing of the shoots. The explants were then transferred into 250 ml glass jars that contains 30 ml of routing media that has the salts of the MS medium, 3% sucrose, and 0.7% agar-agar. And the next step is the cultivation of seed-derived plants. Seeds from the 4 types of chili peppers studied were germinated in vitro on 1/2 MS medium with 1% sucrose and 0.6% agar-agar. The last and final step would be the acclimation and hardening of regenerated seed-derived plants in the greenhouse. Seedlings grown from seeds that reach lengths of 3 to 4 cm were taken from the culture media after the plantlets regenerated in vitro. Roots were immersed in a fungicidal solution for 3 minutes after being washed with running water to remove the residue from the culture media. Plantlets and seedlings were then transplanted into 6 kg pots containing peat moss, covered with perforated clear polyethylene bags and placed in a greenhouse under a black mesh cover. The plantlets and seedlings received 600 ml of solution containing 1 gram of Miracle-Gro and 0.5 gram of phosphonitrate twice a week. The expected outcome of this experiment is that for the in vitro plant regeneration of several capsicum species, an efficient and reliable technique based off direct organogenesis was established. Plant regeneration efficiencies varied depending on genotype, culture medium, and explant source. So that ends my presentation on the in vitro propagation of chili peppers, Capsicum frutescens. Thank you for listening, and as John Steele Gordon once said, and I quote, "Chili is much improved by having had a day to contemplate its fate." Thank you.